Assalamualaikum to all my YouTube students. It's Regal Biology with Khadija, and today we are going to discuss about a very important mechanism by which pathogen can cause disease. The pathogen's mechanism of action by which it can disease in host cell. The very well-known mechanism of secretion, that is Type III Secretion System. We will discuss about the mechanism in detail in this video. So let's start.
Many secretion systems that can be used by pathogens to cause disease in their host cell. Type III secretion system is very well known, so we will dive into the mechanism of bacteria by which it can cause disease in the host cell. In Type III secretion system, the prokaryotic cell, the bacteria will just firstly detect the presence of host cell, that it is present or not. After detecting the cell that host cell is present, the bacteria will form a pore complex and send a effector protein into the host cell. Effector protein can also be called as a virulence protein. It has the ability to cause disease in a host cell. How? It is still unknown. But it is suggested that that virulence protein will suppress the plant immunity. How it will suppress the plant immunity? It is still unknown and out of the question. And after suppressing the plant immunity, the bacteria will start to grow into the xylem vessels of the plant. And you know, in xylem vessels, water flow from roots to the whole parts of the plant. In this way, by the transpirational pull of water in xylem, the bacteria will move into the whole plant and cause wilting and death of the plant. Let's just discuss again these steps by the elaboration of a video. This is bacterial cell, and needles that can used by bacteria to send effector protein into the host cell is present all over the surface of the bacteria. These needles are used to pass, like a syringe is used to pass some material from one place to another place. In the same way, this needle will detect the host cell and then will transfer the effector protein into the host cell. The protein can be called as virulence protein. This protein can, after entering into the host cell, suppress the immunity of the host cell. This is the protein that is entering into the host cell, and there it will suppress the immunity. And after suppressing the immunity, the whole bacterial cell will enter into the host cell, and start to replicate into the host cell. And this is the structure of needle that is present in bacterial cell. This area in a rectangle is a needle that is present in a bacteria. This is the inner membrane of bacteria, and this one is outer membrane of bacteria. And in inner and outer membrane of bacteria, this needle is present. This needle will detect the host cell, and after detecting host cell, it will actually make a pore complex. The pore complex will form after detecting the host cell, and this is pore complex. It will form after detecting the host cell, and when pore complex will form, a effector protein, that is a disease causing protein or virulence protein, will enter into the host cell. This is effector protein, which is virulence protein. It has the ability to cause disease in host cell. How it causes disease in host cell, how it will suppress the immunity of the host cell, it is still unknown. And after forming a pore complex, it will enter into the host cell by the use of ATPase enzyme and enter into the host cell. This is effector protein. After entering into the host cell, it will suppress the immunity of the host cell and helps bacteria to colonize in host cell and to replicate in host cell, to increase number of itself in host cell, so that they can cause disease in host cell. This is the whole mechanism of Type III secretion system in which needle is used. This is needle.